Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 3rd March 2021. As you know, along with the theory we go hand in hand with the practicals also, as you need to perform practicals and the weightage is 30 marks. And one of the experiments that you need to perform is observing the permanent slides and identify those slides and write important characteristics or features of that slide and draw the diagram. So to increase that efforts towards that side, we are going to do in this session the experiment called animal tissues experiment, or either plants or animal tissue experiments. So what is the aim of the experiment? To study one-of-a-kind tissues and variety in sizes, shapes of animals and plant cells such as guard cells, palisade tissue, parenchyma, sclerenchyma, collenchyma, phloem, xylem, squamous epithelium, mammalian blood smear and muscle fibers through permanent slides. Okay, and background of this experiment is related theory, tissues. Like all of you know, what is a tissue? A tissue is an assembly or cluster of similar cells that perform the definite function that's similar in shape and size. Tissues can be classified as simple tissues and complex tissues. Simple tissues, it consists of only one type of cells. They are parenchyma, collenchyma and sclerenchyma. Whereas complex tissues, they possess more than one cell type. So there are two types: meristematic tissues and permanent tissue. Permanent tissue, it can be further classified as simple permanent, complex permanent and of course special tissues. So basically all these are because of the cell. So what is a cell? Is this structural and functional unit of a living body. And what are required for this? Permanent slides we require, okay, to see these slides. And then what is that we require? The materials, many chemicals are required. We use all these chemicals and make permanent slides. And another thing, once we get this permanent slide which is available in our lab, we go to the procedure. Bring some fine, green, fresh and younger branches from a locally accessible woody plant. Thickness must be for a toothpick. Snip the twigs into smaller bits of 0.5 centimeters long. Put the pieces of twigs to the beaker holding water. Boil it for 10 to 15 minutes till the sample settles down at the base. And this way the air inside the sample will be removed. Then transfer the material into a beaker having maceration fluid. Boil it until it turns pulpy and soft for at least 10 to 15 minutes. Put muslin cloth to the beaker's mouth. With the tap water, rinse the material continuously to remove the traces from maceration fluid. Now add some drops of saffronin to the material to color the xylem or the phloem, that is cotton blue. Place the stained material in a drop of glycerin into a glass slide. Split the cells using 2 needles. Plant a cover slip on the slide and observe under microscope. Sketch and compare your observation with the diagram given. So since you are going to perform this, you can write in the past tense. And analysis, normally we take up 3-4 TS of nerium leaf, TS of lotus petiole, VS of root and shoot, of course that much. And what do you observe once we place it? Chlorenchyma is parenchyma cells with chloroplasts. They may have spongy or loosely arranged cells or palisade columnar cells compactly allied. If there is presence of large intercellular spaces into the cells, it is aerenchyma. The protective tissue or the epidermis is the parenchyma tissue forming the outer covering of the leaves, stem and root. Observe the section of the leaf. Mesophyll of the leaf is covered by a lower and upper epidermis. lower epidermis produces small pores known as stomata. In some plants, both lower and upper epidermis of the leaf show stomata. When we are observing under high power as well as low power, we can see the presence of stomata both in the upper epidermis as well as lower epidermis. Tissues are observed for their characters, position in the different parts of the material of the plant. Draw the diagrams and label it. Now put the observation in the tabular form. Like properties, cell wall. In parenchyma, cell wall will be thin. In sclerenchyma, it's very thick. In collenchyma, it is thick at the corners. Then intercellular spaces, many, collenchyma absent, sclerenchyma, collenchyma absent. Nature of the cells, they are living with the presence of nucleus. These are celled, absence of cellular contents. Living nucleus is observed. Shape, oval, polygonal, spherical, rod-shaped or rectangular. Here elongated, variable shape. Cell arrangement, they are arranged loosely in parenchyma. In collenchyma and sclerenchyma, that intercellular gaps will not be there. They are tightly packed. Vacuoles will be present, absent and absent, compactly arranged. So there will be some viva questions related to this also. What are the types of animal tissues? Of course, we said just now. Then what are the types of plant tissues and the permanent tissues like meristem? 3 types of meristem will be there: apical meristem, lateral meristem, intercalary meristem. As the name suggests, apical means apex. It is found in the growing tips of stem, root and helps to increase the length of the plant. Lateral meristem, lateral means side. This meristem is made up of cells that mainly divide into one plane and cause the organ to increase the diameter and growth. Intercalary, this meristem is found in between the permanent tissues. So this is how your external examiner will be asking you